In this video, we will discuss about some of the strategies to improve NK cell specificity. And this area is somewhat similar to that we have discussed uh, in our previous video, in which we have discussed different type of or different generations of this CAR. So we will slightly uh, revise that part of the previous video in this video also. So we will first start with this first generation CAR. So how it can improve the NK cell specificity is by developing the antigens that is not limited to this HLA, that is human leukocyte antigens, and it can be any tumor related antigens that can be targeted via these CAR T cells. And in this case, the intercellular signaling pathways is CD3 zeta. It will cause the release of the cytokines that can kill the tumor cells. It is specific to the antigens that are expressed on these T cells. And after that, in order to improve the efficiencies, people have made the second generation CAR molecules. In this case, what they did is they included another co-stimulatory domains such as 2b4, DAP10, DAP12 along with the CD3. So that will release more cytokines, and that can increase the efficiencies of the CAR cells to target the tumors which is specific to the antigens that are expressed on these CAR cells. In the third generations, what it is, they enhance the signal not just with two co-stimulatory domain but multiple co-stimulatory domains. That is, for example, in this case they have used CD28, 41BB and CD3 zeta, three co-stimulatory domains to target the tumor cells. So this is third generation CARs, and one of the disadvantage could be that it can also affect the neighboring healthy cells nearby, neighboring healthy cells or tumors neighboring healthy cells. Next one is this multi-specific targeting strategies. In this case, the targetings can be done against the ligands that are expressed on the cells. For example, in the myelonemia cells, there is NK. These multi-specific targeting strategies is that they can target two different things. First one is the CD8 that is expressed on the myelonemia cells. This is examples, and second one is this. This CAR can also target this NKG2 ligand which are expressed on the tumor cells. So in this case, the this kind of CAR can target two kind of tumor cells. First one that can express NKG2 ligand, and another one that can another tumors that can express CD38. So in this case, this kind of CARs can work on the multiple antigens that are multiple tumor antigens that are present within the patients. And it can also enhance this ADCC, that is antibody dependent cellular cytotoxicity, because it has this CD16A which is non-cleavable. And the other one is, in order to further increase the specificity of these NK cells, they have also introduced this logic gate CAR. In this case, they have the two kind of CARs. First one is the activating CARs, second one is the inhibitory CARs. So these inhibitory CARs are related antigens are present on the healthy cells, whereas these activating CARs containing antigens that are present on the tumor cells. So when it interacts with the tumor cells, the NK cells gets activated, and it will lead to the death of the cells. That is, example is shown here for AML blast and leukemic stem cells. But when they interact with the healthy stem cells, this inhibitory CARs interact with the EMCN, and thereby does not triggering the release of the cytokines by these CAR cells, NK CAR cells. So in this way, it will further increase the specificity that by targeting only the tumor cells but not to the healthy cells. And other one, last examples here it is shown here. Last example shown here is cytokine armoring. So in this case, what will happen is that how much this how much of the antigens that are or tumors that are present on the acellular domains will determine the payload or the autocrine cytokine secretions. Autocrine cytokine secretions, and this will also helps in improving the its NK cell functionality and also its in vivo persistence. And so this is one of the examples, that is cytokine armoring, where if the tumors are present, then these signals will get triggered and will also cause this autocrine cytokine secretions, that is the this these activating signals will release the cytokines that will further accelerate they will further accelerate the release of the another cytokines that can trigger apoptosis to the tumor cells. This is known as, this kind of secretions are known as autocrine secretions. The other kind of secretions, which is quite opposite to this, is paracrine separations. In this case, in paracrine secretions, the cytokines from the neighboring cells helps in the upregulating or releasing of the cytokines that can target the cells. So, but in this approach, the autocrine cytokine secretions is enhanced once the signaling get activated, thereby increasing the payload of the cytokines related to the tumor's interactions with this with this CAR construct. That's all for this video. If you need to understand more about their different kind of strategies or NK cells, uh, different kind of NK cell generations, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.